Hello, my name is Ariel Rokem, and I'm at the University of Washington, and I'm here to introduce the educational course called Tractometry, Peering into the White Matter. The objectives of this course is for you to learn and understand the basic concepts of tract-based analysis of the human brain white matter. During the course, we'll introduce you to a variety of software that segments white matter into different major bundles, and you'll be able to compare different approaches to statistical analysis of white matter tracts. Our goal is also for you to be able to critically evaluate tract-based analysis applied to a variety of research questions, and to be able to say what you can measure and what cannot be measured with this approach. The brain is composed of networks that comprise multiple brain regions, and the brain regions are connected to each other through connections that traverse the white matter and are composed of bundles of white matter axons. And we now know that the properties of these white matter bundles control complex behavior. They evolve throughout development and through learning, and that the brain properties inside of these white matter bundles are closely related to brain health. We've known for a long time already that brain connectivity is important for a variety of behaviors. These 19th century neurologists already knew that if particular connections between brain areas are disconnected, then that affects particular behaviors, lending to the theory that particular connections are important for particular behaviors. But since the 19th century, we've learned a lot more about the biology of these connections. And we now know that the white matter that comprises the internal portion of the cerebral cortex contains the axons of neurons whose cell bodies reside in the cortex and in subcortical regions. And the white that lends the white matter its color is primarily made out of myelin sheath that wraps around the axons and provides electric insulation that provides lets the axons transmit their information more rapidly from one location to the other, and also allows them to transmit it in a more accurate and precise manner. So white matter is important for rapid information transmission across large distances, and also more accurate information transmission. In addition, the fact that the axons are wrapped in myelin sheaths makes it so that information transmission from one region to the other is less metabolically costly. And that's because instead of that allows for saltatory transmission from one node of Ranvier, these nodes between the myelin sheaths, to another node of Ranvier. Now, in addition to these facts about white matter and its importance in information transmission, we now understand that white matter evolves throughout the lifetime. And we understand through research over many years that this development happens in waves, that there are changes to the white matter that happen in waves that occur at various phases in development and aging. And these phases are phases in early childhood and adolescence. And then in aging, there is the opposite of deterioration of white matter properties. And we now know that these changes in white matter properties that occur in these various waves are related to particular disorders. So neurodevelopmental disorders can be a particular consequence of things going wrong during myelination in early childhood. Psychiatric disorders are related to things going wrong in myelination during adolescence and childhood. And then neurodegenerative disorders affect the white matter in aging and cause the degeneration or demyelination. Now, how do we measure these brain properties? It's very hard for us with MRI to measure the properties of individual axons, and that's because they are very, very small. But the axons, as seen here in this microscopic image, the axons travel through the white matter in large bundles that comprise many, many axons. In this image, postmortem image, looking at a dissected brain from below, where the temporal lobes have been removed, you can see the optic radiations traveling from the LGN to the primary visual cortex. And you can see that the axons in the optic radiations travel in large fascicles or large bundles of axons. And it's the properties of these large bundles that we measure with diffusion MRI. Diffusion MRI takes advantage of the properties of water inside of the tissue, and primarily of the fact that in some places, diffusion of water, random motion of water molecules is isotropic. In particular, in parts of the brain where restriction of the water in all directions is similar, such as in the ventricles, where the water can diffuse freely in all directions, or in round compartments inside of the brain, diffusion is isotropic. It is the same in all directions. But inside of the white matter bundles, diffusion is anisotropic. Water can diffuse more readily along the length of the bundles than across their boundaries. And this anisotropic diffusion is even more restricted in places where there are dense, tight wraps of myelin around the axons. So more myelination leads to more anisotropic diffusion. To make the measurement sensitive to the isotropy of diffusion, to how much diffusion there is in any particular direction, directional gradients are applied across the brain tissue. And without going into too much detail about the diffusion MRI sequences that exist, I'll just say that applying a diffusion gradient along one direction makes the measurement sensitive to diffusion in every voxel to diffusion along that direction of the gradient. And then applying another diffusion gradient here, anterior, posterior, changes completely the image that we see, particularly in parts of the brain where diffusion is anisotropic, such as in the corpus callosum that connects between the hemispheres. And so diffusion MRI gives us an image that is sensitive to the direction of diffusion locally, or to the degree of diffusion in every location in the brain in different directions. And we can take those measurements that are done in different directions and combine them together in order to get estimates of diffusion properties or diffusion tissue properties inside the brain. For example, we can assess the mean diffusivity in every voxel. As you can see here, mean diffusivity is high inside of the ventricles in the center of the brain, where diffusion is not restricted by cell membranes or by other compartments. In the white matter and the gray matter, mean diffusivity is small, and that's because diffusion is restricted by the presence of tissue compartments. Fractional anisotropy is a number between 0 and 1 that tells us about the degree of anisotropy of diffusion in different places. In this image, dark colors are lower numbers and brighter colors are higher numbers. And so you can see that, for example, inside of the corpus callosum, diffusion is very anisotropic. It's very restricted in some directions and less restricted in other directions. And that's because of the presence of myelinated bundles of axons that cross between the hemispheres. In addition to the mean diffusivity and fractional anisotropy, we can estimate the principal diffusion direction, the major direction of diffusion. Here, the major direction or the principal diffusion direction is encoded by color. So if the principal diffusion direction is primarily right to left, that's encoded by red. If it's primarily anterior posterior, that's encoded by green. And if it's primarily superior inferior, that's encoded by blue. And that's a convention that's used to color these kinds of images. And you might see this in other talks today or in some of the literature on the topic. So for example, in the corpus callosum, we see a lot of diffusion right to left, and those voxels are colored here in red. Now, in tractometry, we'd like to assess the tissue properties within white matter pathways. And the first step in doing that is computational tractography. In computational tractography, we put together the diffusion properties along the major bundles in order to assess the location of the major bundles within an individual. Let's see how that works. So we'll zoom in this individual image on a particular location in the brain here in the frontal cortex. If you zoom in on this location, we now model the diffusion in every voxel as an ellipsoid. So in places where the ellipsoid is particularly sharp, that means that the fractional anisotropy is high. Diffusion is very selective to some directions over others. Where it's more round, that means the diffusion is more isotropic. Indeed, here in the ventricle, and the anisotropy is very, very low. And then the color of these ellipsoids that are drawn here on each voxel indicate the principal diffusion direction. So for example, you can see in the corpus callosum, it's red, and that means there's the principal diffusion direction is right to left. In order to do computational tractography, we place seeds, locations in which we start tractography. And then from those seeds that we place inside of the white matter, we propagate streamlines that treat the principal diffusion direction as vectors and propagate a streamline through this vector field. So, for example, if we place a seed here in the white matter in the frontal cortex and propagate it, it might follow the principal diffusion direction through the corpus callosum to the other side of the brain. If we place a seed in the white matter voxel right next to it, it might travel to the back of the brain. If we repeat this process by placing these kind of seed locations in multiple places in the white matter, all over the white matter, we get an image like the one on the left here, where streamlines are filling the entire volume of the white matter. Now, in order to identify specific white matter bundles, we might place regions of interest in particular locations in the brain and ask to select only those streamlines that pass through these regions of interest. For example, in this image on the right, I've placed a region of interest through the corpus callosum and asked to get back from the collection on the left only those streamlines that are in the frontal cortex that traverse through this the corpus callosum ROI that I've placed. So you get one kind of bundle out of this process. And we can repeat this process placing regions of interest to select different bundles and identify many of these anatomically defined major bundles. One way that this has been implemented is called automated fiber quantification, or AFQ, first proposed by Yeatman et al. In 2012. This follows a process very similar to the one that I described before. First, we conduct whole brain tractography, generating streamlines from every voxel in the brain, following the directions of diffusion in all voxels. After that, we place regions of interest that correspond to the trajectory of one of the major bundles. In this case, the inferior fronto-occipital fasciculus. So we place a region of interest in the back of the brain here, in the occipital cortex, and then another one in the frontal cortex, and select only the streamlines that pass through both of those regions of interest. We could do some refinement using a fiber probability map, and then remove fiber outliers to get this kind of clean inferior frontal occipital fasciculus, here also depicted in this animation on the left. Over the last few years, we've created a new implementation of this idea that we call PyFQ. It's open source software that you can download and use. It implements state-of-the-art methods for tractography, for bundle segmentation, for modeling of voxel properties. And it uses and augments the DiPy, diffusion imaging in Python software. And this is a collaboration with, this was led, the development of this was by John Kruper here at the University of Washington, as a collaboration with Jason Yeatman, and also with Eleftherios Garifalidis' group at Indiana University, who's the leader of the diffusion imaging in Python project. And the paper that describes this software was the first paper that was accepted into Aperture, the OHBM journal, and you can read what we found about the reliability of this process. So here are a few examples. This is segmentation of the arcuate fasciculus on the left and the right hemispheres in one individual. Here's a segmentation of the corticospinal tract. And I've also shown you the inferior frontal occipital fasciculus. And the software can segment several other major bundles. Now, after we've segmented out bundles, we can perform what we call tractometry. That is, we can assess the properties of the white matter bundle along its length. So we do this by placing points along the length, 100 points in our case. We sample the bundle along its length 100 times. And then in each location, we gather the FA based on the location of the streamlines in that location. And so at the bottom of the corticospinal tract, we might find the fractional anisotropy, or FA, to be something like 0.6. Then we go and we sample in the next point and the next point and so on until we get what we call a tract profile. This is the properties of diffusion, in this case, the fractional anisotropy, along the entire length of the bundle. And as you can see, the fractional anisotropy varies along the length of the bundle. At this point, I'd like to take a little pause and ask, what can we measure with the diffusion MRI? One way to think about this is by way of analogy. This is based on a Colby paper from 2012 who used a similar analogy. Let's say that you're looking at a map of Seattle and the University of Washington campus where I work, And you'd like to know the properties, for example, how much traffic there is on small streets that are within the University of Washington campus or around this neighborhood. If you measure very, very close at high resolution, you can get an assessment of the properties, for example, the traffic along the roads on campus. But if you measure at lower resolution, you might not see the individual pathways. Instead, you start seeing only major routes through the city, and you get an assessment of the traffic in these different routes. A similar process happens in diffusion MRI, where we're not measuring the properties of every individual axon, every pathway that connects between different parts of the brain. Instead, we're assessing the properties of major highways, these major bundles that contain millions and millions of axons. You might think of this as a really zoomed out version of the assessment of traffic. Now, just because you're not measuring the traffic from my house to the University of Washington campus, but instead get this rough assessment of the traffic along major highways, doesn't mean that this information is not useful. Knowing how much traffic there is through downtown Seattle on the I-5 is useful information. Similarly, knowing the properties of major bundles can be very useful, especially as we find that the variability between individuals and changes along the lifespan are systematic in these properties. It just means that you can't necessarily measure everything that you would like to measure, like, for example, how much traffic there is from my house to the university, or what is the connection between one gray matter vertex in the brain and another gray matter vertex on the other side of the brain. And that's because as a streamline goes through the brain, it might go into one of these major highways, and then it would be very hard to follow it. So here's an example of this. This is with the Human Connectome Project dataset, the 1,041 subjects that have a full diffusion MRI dataset in 24 different major bundles. And you can see that there are there's systematic variation along the length of many of these major bundles. On the left here is the fractional anisotropy, and on the left here is the mean diffusivity in these 24 major bundles. And the systematic variability suggests to us that it's worth looking along the length of the bundle instead of, for example, averaging the properties of the entire bundle. Because if, for example, you were to take the superior longitudinal fasciculus here, and you would average this, you would miss the fact that it goes up and then down and then up again and then down again. Now, I should emphasize, and you might hear from others here in this course also, that there are many names for tractometry. We've used the name tractometry. I've used the name automated fiber quantification, or AFQ, but others have also proposed other names. For example, this wonderful acronym, Pointwise Assessment of Streamlined Tractography Attributes, which actually preceded the AFQ method by a few years, proposed by Derek Jones and colleagues in 2005, PASTA. And this is an image from their paper. Others have suggested the long tract statistics, which does communicate very well what we're looking at here. Here's an image from their paper, and indeed this analogy to the map and the traffic that I've used before. And then others have called tractometry bundle analytics. In fact, Bramschandio et al. Published a paper in 2020 that I also collaborated on that extends tractometry to also include the geometry of the bundles. Here's an image from that paper. Now, once you get these tract profiles, you want to analyze them. There are many approaches for the analysis, and some of them you'll see in some of the talks later in this course. For example, you can do point-by-point comparisons between groups, for example, as done in this paper by Alessia Sarica et al. In a sample of patients with amyotrophic lateral sclerosis, ALS, and a matched control set to show differences in particular tracts. You can correlate the properties with behavior, as done, for example, by Libby Huber et al. In this paper, correlating properties of certain tracts with reading ability. Others have proposed using linear mixed effects models for group and individual comparisons. That an example of that is in the Bramschandio paper that I just mentioned. And then recently, Nathan Muncy et al. Proposed to use generalized additive models, which use smooth statistics along the length of the tracts, and seems particularly well suited to analysis of tract profiles. Finally, some have proposed multivariate or machine learning approaches. We've proposed that in a paper by Adam Ritchie Halford published last year. Maxime Chamberlain and others have proposed similar or and other approaches to multivariate analysis of these data. Now, if you'd like to start using these approaches, one possible way to do this is to use open data sets that exist with these kinds of measurements. There are many, many measurements out there that you can already access openly. I mentioned the Human Connectome project, approximately 1,200 subjects that you can already access. If you're interested in aging and changes in myelination and aging, you can look at data from the CAMCAN, Cambridge Center for Aging and Neuroscience data set that has measurements from approximately 700 individuals. The Developing Human Connectome project has measurements from infants, neonates, and they've measured from approximately 800 individuals, and you'll hear more about neonatal and infant imaging later in the course from Maraki Groth here. If you're interested in children and adolescents, there are several data sets. The Child Mind Institute is collecting the Healthy Brain Network data set that will include 5,000 measurements from 5,000 children and adolescents. And the Adolescent Brain Cognitive Development project will collect longitudinal measurements from approximately 10,000 children and adolescents, pretty massive data set. Finally, the UK Biobank is going to make measurements from approximately 100,000 individuals. And we've already started analyzing some of these individuals. You can also see a poster that John Cooper has done in this poster. In particular, I'd like to focus on the Child Mind Institute's Healthy Brain Network, and that's because one of the barriers to using this data is that it needs to be processed, it needs to be quality controlled, and that for a data set of this size, that can be pretty cumbersome and hard for people who are only getting started in this field. And so what we've done is to create an open, analysis-ready, and quality-controlled data set from the Healthy Brain Network Diffusion MRI data set. From the raw data of approximately 2,700 individuals, we curated a BIDS data set. We did some pre-processing using QSI prep, state-of-the-art pre-processing for diffusion MRI. And then we did, we had experts QC, quality control, a small sample, and through a web app, we had a community, a large community of individuals do quality control, manual quality control, the large part of the data set. And then we scaled that up to the full data set, more than 2,100 subjects using a neural network that learned from these community QC how to emulate the experts. And so we got a very accurate expert level, almost expert level accuracy and quality control of more than 2,000 individuals using this process. And you can access this data through the Amazon Web Services Open Dataset program at this URL. In fact, you won't even need to run more software in order to get to the track profiles, because the track profiles using PyFQ, derived using PyFQ, are already part of that data set. So here's a plot that demonstrates that there is indeed an effect of different quality control levels on these track profiles, that in fact, data of different quality has different track profiles. And so it's important to take into account these quality control scores that we created in this project. Okay, and now, if you're ready, I'll introduce the speakers that will be giving the rest of this course. First of all, Anastasia Indiki from MGH Harvard, who will elaborate a lot more on approaches to finding bundles above and beyond the approach that I showed here. And we'll show some software that does that using both supervised and unsupervised methods. She'll go also into a bit of a survey of the different bundles that you can find in human white matter. Frank Yee will expand on quantitative analysis of diffusion MRI data, providing a little bit of an alternative to this tract-based approach. Thijs Dollender from the Murdoch Children's Research Institute will present other approaches to modeling diffusion in the white matter, in particular pixel-based analysis that takes into account the possibility that in one voxel there might be more than one direction of diffusion, maybe multiple crossing fibers, an idea that he will introduce in his talk. Maxime Chamberlain will talk about the multivariate analysis approaches, or some specific multivariate approaches that he's developed and that I alluded to in my presentation when talking about analysis approaches. Marika Groth here will talk about specifically about application of tractometry to infant brains. And Sila Gench from Cardiff University Brain Research Imaging Center will talk about the application of tractometry in children and adolescents with a particular focus on the strength of longitudinal data sets. And so those are the instructors of the course and the things that they will talk about. Finally, I'd like to introduce myself and my co-organizer, Catherine LaBelle from the University of Calgary, who together with me organized together this wonderful group of instructors for this course. I'd like to thank all the funding support and institutional support for my work. If you'd like to learn more about the work of my group, you can visit neuroinformatics.uw.edu. I hope you enjoy this course.